Hello everyone and welcome. In this video we're going to be checking out 9700 variant 2 of October-November 2018. We'll be walking through it and answering each question step by step. If you want to skip ahead to any question, there are timestamps available in this video and in the comments down below. Also, if you have any questions, whether in AS or in A2 biology, you can write them down in the comments and I can help you answer those. And if you have found any resources that have helped you study and ace your tests, please make sure to share those with the other students that are on this channel. Now let's get started. Question number 1. What's the diameter of a typical prokaryote such as Streptococcus? So you have to know that the diameter of a typical prokaryotic cell is between 0.5 to 0.5 micrometers to 5 micrometers. And that's why we should choose something in that range. Here we have the option 7.5 times 10 to the power of 0 micrometers, which is not within our range. And here we have 75 micrometers, which is obviously way out of our range. Now 7.5 times 10 to the power of 2 nanometers, if you want to figure what it is in micrometers, then you have to know that 1 nanometer is the equivalent of 0.001 micrometer, or in other words, 1000 nanometers are 1 micrometer. So what you have to do in order to convert is basically divide this by 1000, meaning you're going to cancel out this 2 and you're going to end up with 1 decimal place to move. 0.75 is within your range. That's why the answer here is B. Okay. Number 2. A specimen of plant tissue is first observed under a microscope using red light with wavelength of 650 nanometers. The same specimen is then observed under the same conditions, however, using green light with a wavelength of 510 nanometers. What happens to the magnification and resolution when using green light compared to red light? Well, we know that the resolution is going to increase, and that's because we have decreased the wavelength, and we know that resolution and wavelength are inversely proportional. Resolution equals to half the wavelength. So basically, that's inversely proportional, as I have mentioned. And so you're going to look at this and you're going to say, oh, but you're halving 650, and that's going to be greater than the half of 510, and hence the resolution actually is decreasing, not increasing. But that's wrong, because we know that we're not looking exactly at the number when it comes to resolution. Resolution is the minimum distance between 2 points that you're able to visualize. Say you have 70 S ribosomes. Their distance is 18 nanometers, right? So distance between these 2 points is 18 nanometers. When you're observing this ribosome, you have to use an electron microscope with a resolution of 0.5 nanometers. 0.5 means basically the smallest distance between any 2 points is 0.5. So say you have a particle that's as small as 0.5 nanometers, you'll be able to visualize it. However, any smaller than this, you cannot see it. Any bigger, yes, you can see. So 18 nanometers in diameter will be visualized pretty clearly. However, obviously using that microscope, this is not correct, So the smaller the value of the resolution, the greater it actually is, And so that's why I say resolution increases. However, magnification is going to remain the same, because magnification has nothing to do with wavelength. Magnification is only calculated by, multiplying the magnification of the objective lens being used by the magnification of the eyepiece lens. So it has nothing to do with the wavelength. Let's move on. And number 3. Which structures are present in Vibrio cholerae? In Vibrio cholerae, we have circular DNA. Vibrio cholerae, remember, it's a bacterial cell. We do not have mitochondria because these are membrane-bound organelles. However, we do have ribosomes, but not 80 S ribosomes. We have 70 S ribosomes. So that's letter A. 4. The removal of plant cell wall by enzyme digestion leaves an intact membrane. Leaves an intact membrane-bound structure called protoplast. Which statement explains why protoplasts are easily damaged? So we understand that they're easily damaged because the cell wall has, this protective role, basically prevents bursting, because maybe high intake of water maintains the turgidity of the plant cell. But now you've removed the cell wall, so you're not being able to maintain turgidity, and you're going to burst when you gain water. So let's see what are the options. Mitochondria stop producing ATP to maintain membrane integrity. This has nothing to do with it. Net movement of water is no longer limited by turgor. Or secretory vesicles are unable to fuse with the cell surface membrane. No, they're capable of doing that. The shape of the cell structures is changed by the loss of the cell shape. No, cell structures will not be affected. However, what will happen, as they have mentioned here, is that basically there's no movement. The movement of water won't be stopped. This is why the answer here is B. Which statements about are correct for a green plant? ATP is produced by mitochondria. ATP is produced by chloroplasts. ATP forms part of DNA. ATP is produced by mitochondria. ATP is produced by chloroplasts during light-dependent stage of photosynthesis. However, ATP does not form part of DNA. Okay, that's 1 and 2 only. If you know the structure of a DNA nucleotide, you'll know it's, a ribose sugar, deoxyribose sugar, and it's going to have this basically, nitrogenase base, okay? However, it's not going to have 3 phosphate groups, it's only going to have 1. And it's not going to be made of ribose sugar, it's not, like some people will confuse this, they're going to be like, okay, but ATP is very similar to a DNA nucleotide. However, it's also more similar to an RNA nucleotide because they're both made of ribose sugar. But that does not mean that it forms part of DNA, okay? Please remember what a DNA nucleotide looks like. Which process occurs in eukaryotes and prokaryotes? Okay, so in eukaryotes, do we have, eukaryotes and prokaryotes. Hydrolysis does happen in both. Mitosis does not happen in both. Because prokaryotes do not divide by mitosis, they divide by binary fission, which a lot of people may confuse for mitosis, but it's not, okay? Binary fission. So no, mitosis does not happen. Transcription, translation, yes, because they do both carry out protein synthesis, so it's 1, 3, and 4. Okay. A sample of a solution was tested for reducing sugar, and the result was negative. Another sample of the same solution was then tested for non-reducing sugar, and the result was positive. Which step in the test for non-reducing sugar breaks the glycosidic bond? Okay, so the step here should be the hydrolysis using hydrochloric acid. So let's look at this. Addition of Benedict's reagent does not break the glycosidic bond, otherwise we would have had a positive result from the start. Addition of sodium hydroxide does not break the glycosidic bond. It's only added to neutralize the acid that's been added in order to carry out hydrolysis. We have to neutralize it, otherwise Benedict's reagent is not going to work. Boiling with hydrochloric acid, yes, heating to 80 degrees Celsius happens in both cases, whether we're adding hydrochloric acid, whether we are not. And that's why the answer is C. Which row correctly identifies 3 polysaccharides? Okay, alpha-glucose straight chain. Okay, straight chain. Okay, we have amylose or glycogen. Alpha-glucose branching chain. We have aminopectin, glycogen, amylose. Okay, amylose is not a branching chain, and so you can only choose between aminopectin and glycogen. And remember, glycogen is not a straight chain. However, amylose is a straight helical chain. Beta-glucose straight chain, that's cellulose. So that's why C. Okay, however, it doesn't need to necessarily mean helical and straight, like in a synthetic state. Like glycogen could possibly be the answer. It's just basically that these two are wrong. Okay, alpha-glucose straight chain, they just mean like the monomer. I understood it wrong at first. It's just the monomer is, alpha-glucose, and it can form a straight chain anywhere, It doesn't have to be a helix, it doesn't have to be branched. Which row describes a triglyceride? Hydrophobic, insoluble in alcohol. Actually, it is soluble in alcohol, that's why letter B is the answer. Which diagram shows where a peptide bond will be formed? Okay, let's look at this. So we have the alpha carbon, we have the carbon of the carboxylic group, nitrogen. This is very correct. Okay, now here, however, this is I think it's very wrong, because what even is this? We don't have a bond between carbon and carbon. It's a carbon and nitrogen bond. And here this is a very weird distribution. We don't do this actually. The H's stay, otherwise we wouldn't have the H on top of the nitrogen when the bond forms. That's why the answer is letter A. Okay, which statements about collagen molecules are correct? Both types of secondary structure occur within the molecules. Actually, no, because both types we have only 2 types that we know of. They're beta-beta sheet and alpha helix. Okay, alpha helix is definitely not found within collagen. Many people will confuse it because it does have the triple helix structure, but this is not a secondary structure, okay? Large numbers of hydrogen bonds stabilize the molecule. Well, yes, this is correct. We have hydrogen bonds, we have covalent bonds that stabilize the molecule. We also have this part, repeat amino acid sequence determines tertiary structure. These 2 are correct. Yes, because we do have glycine at circular intervals. We also have other amino acids that are repeated in order to form disulfide bonds and everything. So yes, 2 and 3 are right. Okay. Then moving on, the protein glutenine gives bread dough its elasticity. The diagram represents a polypeptide of glutenine. What describes the structure of glutenine? Tightly coiled region, and we have a loose spiral, another tightly coiled region. Interesting. What describes the structure of glutenine? So for number 12, we can see that quaternary structure because there are both globular and fibrous regions. This does not mean that it has a quaternary structure. Quaternary means it's made of more than one polypeptide chain. So we can cancel this out. Quaternary structure because they're both spiral and tightly coiled. Okay, no. basically secondary structure because the loose spiral is an alpha helix. Loose and alpha helix do not go together. Okay, tertiary structure because there's different regions form a 3D shape. This is right. Okay. This diagram correctly shows hydrogen bonding between the water molecules. The symbol delta indicates the partial charge of an atom in the water molecule. Okay, first off, in order to know how the hydrogen bond is formed, you have to know that it is hydrogen that carries the delta negative positive charge, and oxygen is what carries the delta negative charge, and that's because of the distribution, of the water molecules. Considering that water, in place of oxygen, hydrogen and oxygen has much more protons than hydrogen. Hydrogen has only 1 proton, however oxygen has 16. And so basically most electrons that are found inside water are going to be attracted to oxygen. And of course it doesn't happen much, but it's like basically such a weak charge. It does form this weak charge, which is why we give it the delta sign. Delta means weak, okay, or partial charge, okay. And so basically this is why, because it has much more protons, it's gonna be attracting much more electrons, giving it a negative overall charge. That's why oxygen is negative. And the opposite things for hydrogen, because considering it's only 1 proton, the electrons won't be attracted to it. It will be attracted to the oxygen, leaving only the protons at the hydrogen bit, giving it its partial positive charge. And let's look at the options. So as we see, this is correct so far. However, when we look at the structure, it's not 2 molecules of oxygen, it's only 1. Okay, it's H2O, meaning we have 2 hydrogen atoms with 2 partial positive charge, 1 negative and partial negative. Okay, this is very correct, the bond between hydrogen and oxygen. Perfect, let's move on. Okay, and aspirin inhibits an enzyme by reacting with an amino acid that forms an essential part of the 3D structure of the enzyme. Part of aspirin molecules binds firmly with the amino acid. What describes the inhibition? Competitive inhibition, non-competitive inhibition, reversible inhibition. Let's look at this. He said that part of the aspirin binds firmly to the amino acid. Okay, and this amino acid forms part of the 3D structure of the enzyme. Interesting. So basically, this firmly does not indicate it's a reversible inhibition. And considering that it binds firmly and it's non-reversible, it's going to be a non-competitive type of inhibition. And that's why the answer is 2 only. Okay, the table shows the Michaelis-Menten constant Km for 3 enzymes. Which interpretation of the information is correct? Okay, so we have statement letter A, enzyme C has a Vmax which is half of that of enzyme B. Let's look at enzyme C. Here he gave you the Km value for each of the enzymes, and based on that we can't really determine the Vmax for any of these enzymes. I don't know why they would basically discuss the Vmax and the time interval at which the Vmax will be reached. You can't really determine that. Enzyme F has the greatest affinity for its substrate. Let's look at enzyme F's Km value. Yes, it does, because here it's a negative sign, meaning that this is a very, very small number. And the smaller the Km value, the higher affinity of the enzyme to its substrates. If you do not understand the Km value, let me explain this to you. So first off, let's look at this graph, okay? It's a graph of substrate concentration, okay, against the rate of reaction. And so basically, we know how it normally goes out. Basically as you increase substrate concentration, it's no longer a limiting factor, you reach a plateau, and that's basically when substrate and enzyme are only added. However, when you add a competitive inhibitor to the mix, you get this type of graph. Okay, you can see that the rate of reaction keeps increasing, but at such higher substrate concentrations does it reach the same maximum rate of reaction known as the Vmax, right? The Km value is known as the substrate concentration at which half the Vmax is reached, or the substrate concentration at which half of the available enzyme active sites are fully occupied. Okay, and it indicates, the substrate concentration needed to find the Vmax. However, it does not tell you what it is, And so basically, here, if we have a lower Km, like in this case, obviously, we know this is half the Vmax, for example, half the Vmax, and this is the Km value. When we add the competitive inhibitor, the Km value increased. Okay, this is in the presence of competitive inhibitor, and this is the new Km, and it has increased. Now you're gonna be like, okay, it's increased, but how does this indicate that there's a higher affinity? Well, let me tell you. When you have a competitive inhibitor, what happens basically is that it distracts the enzyme from the substrate. They're no longer as much attracted to each other because there's a third party member. They're busy talking to it, right? It's like basically when you're with a friend on your own, you're going to be talking much more together than if there was another person coming in between you guys. You're going to be, distracted by them. right? And so the attraction between you two decreases. So in other words, when you basically have a high competitive inhibitor, what happens to the Km is it increases, right? And in the case of a competitive inhibitor, there's lower affinity of you guys to each other, right? Of the enzyme to its substrate. There's a lower affinity, and there's a higher Km value. And the opposite can also be said. When there is a lower Km value, you have a higher affinity. I hope it is clear, and that's why the answer here is C. Let's look at the last option. Enzyme P has a Vmax of 6.0. Again, we cannot determine the Vmax. Perfect. Okay, question number 16. The diagram is a drawing of an electron micrograph of a typical animal cell. How many layers of phospholipids will a molecule of oxygen cross to travel directly from X all the way to the cell to Y of the matrix of the mitochondrion? Okay, in order to travel all of this, how many layers of phospholipids? Interesting. So we know that phospholipids are bilayers usually, okay? And we know basically that phospholipids are what forms the cell surface membrane. And if you know the structure of the cell surface membrane, you know it looks something like this. This is a phospholipid molecule, and these are phospholipids right now, and it usually looks something like this, right? With proteins and whatnot, and is 7 nanometers in diameter, okay? So this is only one layer of phospholipids, this is the second layer, and this is only the first cell membrane, okay? This is cell surface membrane, and then it crosses this one over here, which is also a double layer. So far here we have 2, and that's another 2, and that's another 2, so far if you add all these, you're gonna get 6, and that's only to cross and reach the matrix. How could a water molecule cross the cell surface membrane of animal cells? Water can pass by phosphorylated diffusion and by osmosis. Phosphorylated diffusion is when it crosses using channel proteins. However, by osmosis it can move because it is a very small molecule. Despite the phospholipid having hydrophobic fatty acid in their core, it can cross it because, as I mentioned, it's very small that the phospholipids, they just go like, okay, fine, go in. You're very tiny. We don't care. You're not going to affect us, right? However, if it was a large protein that was, soluble in water, they're going to be like, okay, you get out now. We don't like you, okay? And so carrier proteins, well, actually, no, they pass through channel proteins, okay, and they're called aquaporins, okay, and so that's aquaporins, it's cholesterol molecules. Really, they cannot cross the cholesterol molecules, so that's 2 only. Okay, the diagram shows a xylem vessel in a root and 4 adjacent parenchyma cells. As water moves up the xylem vessel, it is replaced by water from cell D. Water in cell D is replaced by water in cell C, resulting in a flow of water from cell A to the xylem. So which cell has the least negative water potential? If water moves from a region of higher water potential to lower water potential, down water potential gradient, that moves from A all the way to the xylem, that means that basically this is going to have the highest water potential, meaning it has the least negative water potential. Remember, there's multiple wordings. We have high water potential, we have less negative water least negative water potential. We have the more negative, the more concentrated it is. We can say most diluted. You can say basically least concentrated, most diluted, least negative. Okay, all these wordings. Let's move on. Now, number 19. Agar cubes can be used to demonstrate the effect of changing surface area to volume ratio on diffusion. 3 different agar cubes are made using dilute acid, were placed in an indicator solution that diffused into the cubes. When the indicator came into contact with the acid, it changed color. The cubes were 1 centimeter cubed, 2 centimeters cubed, and 3 centimeters cubed, and were left in the indicator solution for 10 minutes. All other variables were kept the same, and the results were recorded as diagrams. The results for 2 centimeters cubed are shown, and we can see here basically we can see that the original color cube changed color to a depth of 0.5 centimeters. Okay, which diagram presents the results for 1 centimeter cubed, 3 centimeters cubed, and so on? Okay, so basically the smaller the number, okay, 1 centimeter cubed is going to have the greatest surface area to volume ratio, okay? And this is like something that you can easily determine. Just the smaller the object, okay, the greater the surface area to volume ratio. And so here basically this is going to be having, of course it's going to have changed its color completely by the time that obviously this has changed color completely, because they're all left for 10 minutes, so it's going to be having changed color completely. However, this is not going to, basically change color to a depth of 0.5 centimeters, okay, or basically, let's look at the other options. Okay, I would assume it's this option, that's right. Okay, the photomicrograph shows the cells in different stages of mitosis. Which statements are correct? Okay, so we have letter P. This is anaphase. Okay, let's look at the numbers. Cell T shows metaphase. Cell T does show metaphase because the chromosomes are aligned across the equatorial plate or the equator of the cell. And number 2, DNA replication occurs in cell R. DNA replication, now let's look at R. R is actually during prophase, and DNA replication happens during interphase, which is before, which is when cell looks like S. So this is wrong. Okay, this is correct. The amount of DNA in cell B is the same as the amount of cells and the amount of DNA in cell T. Let's look at cell T. Cell T is in metaphase, and cell B, as I mentioned, is an anaphase. Okay, so they're both, yes, do contain the same amount of DNA. It only changes when synthesis phase happens, when S phase happens, or when DNA replication happens, which is basically during interphase. So I would say R has double the amount of chromosomes than S, unless this is done with the replication. Okay, if it's in G2 phase, then I would say they are equal, you know? Now, the correct order of the stages is S, R, T, P, Q. Let's look. So S is interphase, correct, and then followed by prophase. T is metaphase, and then P is anaphase, and Q is telophase. And if you are confused with basically the pattern, you have to understand it's first interphase, followed by mitosis, and then obviously cytokinesis, right? And in this case, they didn't mention cytokinesis. However, we know mitosis is basically PMAT. Prophase, metaphase, anaphase, telophase. Okay, and that's why I immediately said SRTPQ. And that's why the answer here is 1, 3, and 4, which is letter C. 21. Which changes in the group of a million cells dividing mitosis would be necessary for the formation of a tumor? Mitosis is no longer inhibited by cell-to-cell contact. Okay. Cells acquire the ability to migrate to new colonies. Well, this is actually involved in metastasis and, building secondary growth and primary growth. So first we know that basically one tumor is going to be formed in a specific place, and then when basically if this tumor is malignant and not benign, meaning it does not travel different places in your body and stays where it is, basically if it is malignant, then the cells will be able to travel. They acquire this ability, and basically they're going to leave, whether through lymph or the blood vessels, and set up new colonies in different places in your body. And that's basically when they start to form the secondary growth, when they settle in another organ and they start to divide by mitosis, forming another growth, which is known as a secondary growth. And this is not required for formation of one tumor; it's a formation of multiple tumors. Okay. And so it's not necessary for formation of tumor, because this is also, as I mentioned, for a specific type of tumor, malignant tumor, okay? And it's not just one tumor again, it's 2. That's why the answer here is wrong. Cells become able to divide indefinitely. Obviously, this is needed, okay? And no longer inhibited by cell-to-cell contact, obviously, because, normally, hormones would inhibit mitosis, maybe something else, like nerves, something, there's lots of messages that come to cells to stop them from doing this. But mostly when it comes to a tumor, the cells are going to be going crazy. They're not going to be responsive to any kind of nervous or, hormonal interaction. They're just going to be dumb, okay? That's why the answer here is letter B. Now, 22. Which row shows some of the events during mitotic cell cycle and correct time sequence? Okay, let's see. The time, and we can see it's passing by. Centromere of each chromosome splits. Interesting. Nuclei become visible. Nucleoli become visible again, so that's at the end, okay? Nuclear envelope breaks into small vesicles. This happens before all of this, actually, so this is wrong, okay? Nuclear envelope breaks into small vesicles, okay? Spindle microtubules are produced. Well, spindle microtubules, yes, they are produced right now. You can say so. Centromeres of each chromosome split. Nucleoli become visible. I say letter B is correct. Okay, and then we have nucleoli become visible. This is again at the end. This is before this. Spindle microtubules produced. Centromeres of each chromosome. Okay. Nuclei become visible. Again, wrong place. Letter B. What is the maximum number of hydrogen bonds in the length of DNA containing 700 base pairs? Okay. The maximum number of hydrogen bonds. We know that we have either 2 hydrogen bonds or 3 hydrogen bonds between each base pair, because it's either cytosine with guanine, C to G, which is 3 bonds, or A to T, which is 2 bonds, right? And so to get the maximum number of hydrogen bonds, you need 3. You need them all to be cytosine and guanine. And he said there are 700 base pairs, okay, meaning that there will be 700 bonds. And 700 times 3 is 2,100, which is letter D. What is the common component of 3 molecules, RNA, DNA, and ATP? They all have an nitrogenous base, okay, they all have phosphate. Let's look at this. Adenosine, no. Okay. Adenosine is just a name. Okay, it's adenosine triphosphate. Hydrogen bonds, not within the ATP molecule, no. Okay, phosphate or ribose. DNA has deoxyribose, not ribose. That's why the answer here is C. Okay, 25. photomicrographs of dicotyledonous plant, root, stem, and leaf are shown. Interesting. It says which row is correct for the labels 1 to 5 of the on the photomicrographs. Let's look at this. So 1, we can look at the xylem, as you can see, and so let's see, here we can label xylem. Okay, number 2, we can look at the phloem. Okay, and number 3, let's look at this number 3. Where's number 3? Yes, number 3, we can see it's endodermis, and these are cells that basically have suberin on them. They have experience strip. Okay, and number 4, where's number 4? Right here, it's the cortex, yes, okay. And number 5, it's the epidermis, and that's what it says for D. 26, which features apply to phloem sieve tube element and to xylem vessel elements. So let's see, no cytoplasm. Actually, phloem sieve tube element has cytoplasm, however, to the side, okay. It's very, to the side. No end walls. Well, actually, it's only applicable to xylem because xylem has no end walls. However, phloem sieve tube has perforated end walls, meaning they have holes in their end walls. They have the sieve plates at the end, okay. No nucleus. Yes, they both do not have a nucleus. That's why the answer is D only. 27. Which statements correctly describe an event in the process of mass flow in the phloem sieve tube elements? Okay, so in the process of the mass flow, let's see. Any part of a plant where sucrose is loaded into a sieve tube element is called a sink. Loaded into a sieve tube element is actually a source, not a sink, and that's why the answer here is not A. Okay, loading sucrose into a phloem sieve tube element increases the water potential inside it. Actually, no, because we know that when you load something with sugar, you're making it more concentrated, right? Like basically when you put sugar into your tea, you're making the tea more concentrated. You're decreasing the amount of water inside, increasing the amount of sugar inside, right? So it becomes more concentrated. It's not increasing the water potential, it's the opposite thing. Okay, mass flow in the phloem is a passive process occurring at the same rate as diffusion, not necessarily as the same rate as diffusion because we have different rates of diffusion, okay? But yes, it is a passive process. Sucrose moves down a hydrostatic pressure gradient in the phloem from source to sink, perfect, letter D. The diagram shows a potato tuber at different stages. Okay, so we have a dormant tuber, not growing, sprouting roots and shoots, okay? And then growing roots and shoots, which is letter 3, growing roots and shoots, and then making new potato tubers. Number 4 is basically making new the tubers. Interesting. So which identifies a stage where the potato tuber acts as a source and a stage where it acts as a sink? The potato tuber itself acts as a source. Let's see. It is a source, I think, during 2, and also basically growing roots and shoots when they are growing. Well, basically we still have leaves, so we're not looking exactly. I think it's number 2 when it's a source, and then number 4 is actually when it's a sink because it's growing new. Yeah, it's perfect. Okay, that's where B. Now, number 29, what describes the role of atrioventricular node of the heart? Atrioventricular node, let's see, it causes the muscles of the ventricles to contract from the apex upwards. Actually, this is not the role. The role is actually to conduct the impulse and basically pass it to the Purkinje tissue. Purkinje tissue is then going to conduct it and cause the apex to, contract. Cause the heart to, the walls of the ventricles to contract from the apex upwards. It delays the transmission of a wave. Electrical activity from the central node. Perfect. Yes, it does do that. Okay, because you just don't want the atria and ventricle to contract at the same time. Central atrial node is going to be sending the impulse or basically electrical activity all throughout the atria walls, and so the atria are going to contract. If the atrioventricular node does not delay the activity, then both atria and ventricle will contract at the same time, which will be very confusing for us. I mean, at least for the it's a new wave of electrical activity in the ventricles and does not initiate anything. That's the role of the central atrial node. It only conducts. It provides a non-conducting barrier. Okay, this is not the role of it. It's actually the role of the non-conducting barrier. Okay, the shows pressure changes during the cardiac cycle, which arrow indicates ventricular systole. Ventricular systole happens at B. It's where at this point where there's a peak in the pressure. Okay, which process can be carried out by a mature blood cell? A mature blood cell can carry out active transport, cell division, phagocytosis, or protein synthesis. Okay, it cannot carry out phagocytosis. Neither can it carry out cell division because it does not have a nucleus in this case. It does not carry out phagocytosis. Just does not contain. It's not a phagocyte. Number 1, okay, and basically active transport. Yes, it can do that. Okay, protein synthesis. It does not have a nucleus again, so it cannot do that because protein synthesis requires transcription. Okay, let's move on. So Hemoglobin can bind to carbon dioxide, carbon monoxide, and oxygen. Which statements about the binding sites of hemoglobin is correct? Remember, carbon monoxide is out of our syllabus. They won't be asking you about this anymore. However, you should know. Let's look at this. Which statement about the binding sites of hemoglobin is correct? Carbon dioxide and carbon monoxide bind at one site. Oxygen binds at a different site. Actually, no. Carbon dioxide binds at a different site, and oxygen and carbon monoxide will bind at the same site. So that is what this says, I think. Yes, perfect. Okay. Which tissues are present in the bronchus? Cartilage, serrated epithelium, smooth muscle. Cartilage is present in the bronchus. Everything here is present. Okay. Cigarette smoke is not part of your syllabus anymore. 35. Symptoms of cardiac obstructive pulmonary disease, again not part of your syllabus. Which statements about infectious diseases are correct? Cholera has a vaccine available and is caused by a bacterial pathogen of Vibrio group. Measles is a vaccine. Okay. Cholera does have a vaccine. We have measles as a vaccine and available and is caused by a pathogen called morbillivirus. That is true. Smallpox was eradicated by vaccination and was caused by a bacterial pathogen called variola. Okay. Now, bacterial pathogen. Smallpox is caused by a viral pathogen. It's a virus called vaccinia and variola virus. And so that's wrong. It's only 1 and 2. Okay. 37. We have the antibiotic tetracycline binds to the small subunit of bacterial ribosome, stopping protein synthesis. Interesting. Bacteria have become resistant to tetracycline because of effective mutations. What effect could produce the resistance to tetracycline? Preventing tetracycline from binding to the bacterial cell wall. Okay. Or preventing tetracycline from entering the bacteria cell. I think changing the shape of the ribosome. Okay, the small subunits at least. Preventing tetracycline from inhibiting transcription. Preventing the production of tetracycline by ribosomes. I think maybe preventing it from entering the bacterial cell, because if you don't, it does not enter, it cannot bind in general. Okay, so on the surface of macrophages, yes, it is found on the surface of antigen presenting cells, which are basically, after the macrophage is going to engulf, any pathogen, it's going to leave the pathogen's antigens on its surface, and that allows basically for clonal selection and clonal expansion and everything, which is part of the primary immune response. Okay, that's in blood plasma. It's not going to be found. Antigens are not just lying around in blood plasma. We have antibodies, not antigens. Okay, that's repeat. The graph shows how the body reacts following exposure to an antigen for the first and second time. Interesting. So what is the direct cause of the increase in antibody production after the second exposure to antigens? The second exposure is because it's caused basically so the direct cause is actually plasma cells. Okay. Vladimir says that many students are going to be making this mistake, and it's actually what I would have chosen at first guess, but because I read direct, I was like, okay, no, we're choosing plasma cells. And that's because the B memory cells, yes, they are going to be necessary for this to happen. However, they are not going to be the ones that are going to produce the antibodies, are they? That's why the answer is C. Okay, why has it proven difficult to develop an effective vaccine against malaria? Okay, so the reason is because it's difficult. We actually have 4 different reasons, but let's read the options. Mosquitoes have many stages in their life cycle. Mosquitoes have many stages in their life cycle. Actually, we're talking here about the pathogen's life cycle. How basically, it's just the pathogen's life cycle. Is it mosquitoes life cycle? it's the pathogen's life cycle. مش هي أول شي راح تكون كمل طيب 40. The human immune system does not recognize the antigens of the parasites. So it does recognize it, however, it does not see it most of the time when it's concealed. The parasites can only be attacked when outside the liver cells and red blood cells or vaccines are rapidly broken down by proteases in the stomach. Okay, the vaccine is not going to be taken in by the mouth for malaria. Okay, so that's wrong. However, this one is alright. Okay, there's lots of antigenic concealment. The pathogen is mostly inside liver cells and red blood cells. When it's outside, the reason it's so difficult to attack it is because it has changed its antigen. It has many, basically every time it goes out, basically it has different stages, I think, in the life cycle, and that's why basically it keeps on changing its antigens, and that's why basically the parasites can only be attacked when outside the liver cells and red blood cells, and I think the answer here is C. I forgot. Okay, you can just look it up. Life cycle of Plasmodium here. See, it has many stages in life cycle. Perfect. Okay, look at this. Yeah, the process contact when it's yes. Okay, so that's letter C. Again, because basically the plasmodium has many stages in its life cycle. And this was it for this variant. Hope this video was useful. Please make sure to share it with other students if you found this video helpful. And also, good luck everyone. Goodbye.